Hi, I'm Mr. Baker, and this is your presentation over Unit 3, Cells. We have a few learning objectives. You're going to tell me about what a cell is and the two types and the cell theory. You're going to describe the structure and function of cell membranes for these first two sections, and the other three are later on. First one, 3.1, all organisms are made of cells. A cell is the smallest unit of life that can function independently and perform all the necessary functions of life, including making more of themselves. It's a three-dimensional structure like a balloon that allow all the essential chemical reactions of life to take place. Nearly all cells contain DNA. Some cells do not, like RBCs, red blood cells, and your hair and nails. Those are cells that their job doesn't require the need for DNA. Most cells are microscopic. However, you can see them in the grocery store anytime you want by looking at the eggs. The largest single egg on the planet is the ostrich egg, weighing about 3 pounds. You will need to know the big five: Hooke, Van Leeuwenhoek, Schleiden, Schwaden, and Virchow. Hooke, who was British, discovered the cell in 1655 under a very old school microscope. He looked at tiny thin slices of cork and he saw the little pores and he says, hey, this looks like the places where monks live, and those are called cells, so ergo the name of cells. Here is a picture of his old school microscope, and for about 1670 that was the best we had. Anton van Leeuwenhoek was Dutch, and he perfected the microscope, and he was the first to see even smaller things like bacteria, also called prokaryotes. This is one that you held up to your eye. The cell theory was generated by these three men, Schleiden, Schwann, and Virchow, and they were all German. The three parts of the cell theory: all living things or organisms are made up of cells and their products. Cells are the basic building units of life, and new cells are created by old cells dividing in two. A few landmarks in cell biology. The bold ones are the only ones that I might ask you a question about. In 1595, the first compound microscope was made by some dude named Janssen, and he was Dutch. 1655, Hooke made the cells. About 20 years later, Van Leeuwenhoek found the bacteria. The cell theory was started in 1839. Virchow added to the cell theory about 20 years later in 1858. And in 1953, Watson, Crick, Wilkins, Franklin, Rosalind Franklin, don't forget her, identified that the DNA structure was a double helix. You will know that there are two types of cells that exist: the eukaryotes, like us, that have a nucleus, and the prokaryotes, which have no nucleus, and those are the bacteria. The bacteria are simple, but there are many types of them. No nucleus. They are smaller, and they evolved first about 3.8 billion years ago. They are metabolically diverse. They can use many different types of energy sources, unlike cells like us, which have to use glucose. Some can live without oxygen. Some use sulfur, like in the deep sea hydrothermal vents. Some use hydrogen, and they also invented photosynthesis, and that's a type of bacteria called cyanobacteria. There's a picture. They're simple. Some of them have tails called flagellum, and they use that to move around. Another picture. Eukaryotic cells have a nucleus, and they have things called organelles. More about those later. The word eukaryote comes from the Greek for true nucleus, karyo being nucleus, and eu for true. There's a picture of a few eukaryotes. They evolved about 2.7 billion years ago, which is 1.1 billion years after the bacteria were first identified. They are not all multicellular like us. Some of them are single-celled, and one large group that contain many single-celled eukaryotes are called the protists, and there's a picture of a few of them. They all have organelles. Here's a picture of animal versus plant cell, and we'll get more into those in a couple sections down. You need to be aware of the endosymbiosis theory, which states that a larger cell engulfed the smaller cell, and eventually they worked out a deal where if you give me a place to live, I'll hook you up with some extra energy as rent. And that's what the little bacteria said, and the big cell said, you know what, that's a good deal for me. And those eventually became the chloroplast in plants and the mitochondria in plants and animals. That's section 3.1. Cells, the early biologists, the cell theory, the difference between prokaryotes and eukaryotes, and the endosymbiont theory are what you're expected to know. 3.2, cell membranes are gatekeepers. The plasma or cell membrane, those are interchangeable. They consist of phospholipids, which have one polar head. Polar means it's hydrophilic, water-loving, and two nonpolar or hydrophobic, water-hating tails hanging down from it. They are organized in a phospholipid bilayer, and they attach themselves head to tail to tail to head. Here's another picture. Proteins and other molecules will be attached to it, and depending upon where they attach is what that molecule is. For example, if you attach to the head, you're going to love water. If you attach to the tails, you're not going to like water. For example, on the right-hand side, the surface proteins are attached to the polar heads. Those are polar molecules. In the left side, you have a transmembrane protein. It goes all the way across the membrane, so it has polar areas, non-polar areas, and another polar area. This slide just writes out what I said on the last slide, that you're going to go to where you attach. And if you like water, you're going to be near the polar area. And if you don't like water, you're going to be near the non-polar tails. There are 4 types. Some of them are receptor proteins, recognition proteins, transport proteins, and some of them are enzymes. The plasma membrane acts as a fluid mosaic, meaning that things can move around. Cholesterol is important because it helps maintain the flexibility of the membranes. For your fluid mosaic, imagine the surface of a lake where the heads and tails are in constant motion and the proteins bob and move around, and there's a structure that you'll see that's a lipid raft, and it's like a sailboat. There's some more detailed information. They also have fingerprints, and this is important for organ transplant. You got to get those fingerprints to match up perfectly, or the host could reject the organ that's being transplanted in. That would be a bad thing. That's a slide telling you what's all you need for quiz 3.2. Study hard, and we'll see you next time.